is so much uncharted territory in the genome. We have the list of genes, but we don't really know where they belong in their relationship to one another. It's like having the index to a book, but not knowing what the pages are.
We got the parts list from the Human Genome Project. We kind of understand how they interact with each other fairly well nowadays, but we really don't understand the dynamics of how these interactions change, for example, and how they go awry in disease.
Most diseases are not caused by one single mutation or one single change in the DNA sequence of a person, which makes it a lot more complicated for us to find these instances and also to predict them.
Just like a social network, you learn a lot about a gene by what it associates with.
Proteins very rarely function on their own. They often have their friends that are connected to them, and they function in a complex.
In my own lab, I use information like a gene is associated with a couple other genes, and so then we think that mutations in that gene are going to lead to drug resistance, and we can predict that and test it much more rapidly than if we didn't have that other information from the network. We have the ability to make guesses about what proteins might be doing, but it's also critical to really test those, and one of the focuses of my work is to go from guesses to reality. I actually came to the Donnelly and started my lab here because it was the world leader in figuring out these social networks of genes.